Welcome to Biotech Stage. It's a stage or podcast where we talk with different biotech professionals from the globe and also from the whole spectrum of biotech positions, including presidents, vice presidents, directors, team leaders, entrepreneurs, and most importantly, the people who work on bench, that means scientists. Our guest today is Dr. Sourav Majumdar. Dr. Saurabh Majumdar has lots of experience in different companies in India. It's almost like 2 decades of experience. He did his PhD in biochemistry and molecular biology, and he started working at different companies, including USB, Wockhardt, INTAS, and Serum Institute of India. And currently, he is the technical director at BioAgile. And he got a lot of experience in recombinant proteins, their characterizations, and lot of information about biotechnology that we can discuss today. So, welcome, Dr. Majumdar, on this podcast.
Thank you very much for so nice introduction by Dr. Odun. I'm really thankful that I've got an opportunity to speak about my experience on the platform, and really it will be it will be very fruitful for future journey of biotech.
Yeah, thank you. So, Dr. Majumdar, very first thing that I wanted to ask you is you moved from different companies, Intas, USV, Wockhardt. How was your journey in biotech? I mean, you must have faced lot of problems, and you must have overcome those challenges. So, how was your journey? And if you could please share some of the information on that.
Actually, immediately completion after my post-graduation degree in biochemistry from University of Pune, I have joined Wockhardt Research Center. So that research center was dedicated for the insulin and insulin structure and analog. So I was mainly involved into formulations purpose and also the analytical characterization of API. That's mean the active pharmaceutical ingredient. And the structure and analog formulation was the a big challenge because if you see any molecule like other molecule like HGH, IL-2, PEG, CSF, and other things. As compared to that, the insulin formulation was bit difficult because there is no final illusion in the excipients buffer. So the
Mm-hmm.
API was different, formulation buffer was different. We have to mix up. And in during that mix up, you need to do the certain formulation like NPH 30/70, NPH 50/50, like that combinations you have to get insulin in overall in a solution. So it depends on the patient that how the patient and what is the degree of the diabetes, if his diabetes is very high or if he want to sustain that insulin
Mm-hmm.
inside the body. So based on that, the formulation was there. And there during the formulation I was very I will take an example that is a very clear example. During the formulations we are using protamine sulfate. So protamine
Mm-hmm.
sulfate usually form a crystal along with the insulin. But this protamine sulfate is a very small peptide, around 32 amino acids. Out of 32 amino acids, the 28 amino acid was arginine.
Mm-hmm.
And insulin during the process, if you follow that pre-pro insulin to pro insulin and then insulin, so there is using some step called trypsin. So trypsin
Mm-hmm.
was already inside the bulk material. So trypsin active site was arginine and lysine, and the moment we are using the protamine sulfate, that protamine sulfate they easily chew and they are not able to form the crystal formation. Then
Mm-hmm.
we find out that total removal of trypsin from the bulk is very essential. Otherwise, these complex formations or the crystal formation will be not appear in the formulation buffer.
Yeah,
So
okay.
that
Okay.
was the challenge, and then we have detected this presence of trypsin by using an assay, and we have quantified that how much trypsin was still present in the bulk, and because of that reason that kind of formulation is facing that kind of challenges. And insulin itself is a
Ah, okay.
very small molecule, around 5.6 kd, and that 5.6 kd
Mm-hmm.
molecule, if you see that it is a very delicate structure, and to
Mm-hmm.
optimize that part in the formulation buffer is very important. But because glargine is a long acting molecule and it is compared to insulin, and lispro is a very soft acting molecule. So whenever
Mm-hmm.
you are talking about something called glargine, so the glargine itself is a it will sustain in the body, so it
Mm-hmm.
doesn't require any kind of complex formations. It will be in only the clear cut solutions, and it will be solubilized in a buffer. and it will sustain there. The difference between insulin and glargine was that arginine was added in the B chain, and there is one replacement of the glycine in the A chain. So that was the difference. In case of Lispro itself is telling is a Lispro, lysine and proline. So there is an inversion between a molecule between proline and lysine, that is the control sequence of insulin. And when you
Mhm.
are converting in the form of Lispro, so it will be in the form of lysine and proline. There's minimum conversion will change the characteristics of the molecule in a very fast as a fast acting manner. So
Okay.
that was the scenario. So these are the challenges we have faced at initial stage, and then I have decided to move in further for my further growth in USV, where I worked out in IL-2. It's very tricky molecule, IL-2 HGH, and was there. And in
Mhm.
INTAS, I worked with the GCSF and PEG GCSF. Then I went
Mhm.
for PhD. There I also worked out some wild protein, and in Serum Institute of India, then I have joined. Before that, I was with the Vitin, and Vitin I spent 1 year. So they are handling with the glargine molecule as well as bevacizumab. So that
Yeah.
was a good exposure to get is a mAb. And when I was in Serum Institute, I worked with trastuzumab, vastakunimab. Serum wanted to introduce himself in a molecular that is a mAb, and they have already opened a domain. But after
Mhm.
during the corona session also I have worked out in a COVID vaccine. So that
Mhm.
COVID vaccines means it's not a Covishield, it is a Covovax. It's a
Mhm.
recombinant protein vaccine, and currently I am dealing with a small peptide like peptide to pembrolizumab and other maps also, and along with that some synthetic molecule also for analytical characterizations in
Okay.
agile
Okay.
biosciences.
Okay. Okay. Yeah. Thank you. Thank you very much for this information. And as you said, the proteins once you make on a large scale for any commercial production, the characterization is very important. You need to make sure that it is fully biologically active. And you have done a lot of work for protein characterization.
Yes, yes, because
Mm-hmm.
for scale up that process, for example, insulin.
Mm-hmm.
Insulin, if you think about that, glycosylation part is not there. They are exactly processed on E. coli or Pichia or yeast system.
Mm-hmm. Mm-hmm.
But when we're talking about mAb, if you do the scale up,
Mm-hmm.
it will be a huge difference in the glycan profiling.
Yes. Yeah.
Because the moment you do the scale up, that scale up will not proportionally enhance the glycosylation part of that particular molecule.
Mm-hmm.
And
Mm-hmm.
glycosylation is very, very important because you need, that's why we are doing some analytical characterization as well as comparability study between the low scale to high scale, 10
Mm-hmm.
liter, 2000 liter, then 10,000 liter, like that
Mm-hmm.
small scale to flux to fermenter in a high scale manner.
Mm-hmm. Mm-hmm.
Because that glycosylation pattern will change, and there is a possibility of
Mm-hmm.
changing the acidic and basic variants
Mm-hmm. Mm-hmm. Mm-hmm.
because of that reason. So that will impact on the in vivo conditions when it is injecting in the body. That is a
Okay.
receptor mediated interaction. It will be completely different.
Yeah, and that's a very tough challenge for all the biosimilar companies because they need to make sure that their molecule
Yes,
is identical to the innovator's molecule. So something like
yes,
you do
yes.
and your group does is very very important to ensure that it's fully biologically active and all the glycan are placed on the right
Yeah, for
place.
that purpose, yeah, for that purpose, this level of characterization is very important in terms of
Mm-hmm,
primary, secondary, and
mm-hmm,
tertiary and
mm-hmm,
quaternary structure of the molecule.
mm-hmm,
So, in primary level characterizations, what actually we are doing the amino acids composition analysis, peptide mapping, peptide mass fingerprinting, intact mass, subunit mass, glycan, acidic variant, and
mm-hmm.
SEC mols also and SEC HPLC also for aggregate analysis. In similar way, also we have identified the element test for the free thiol
Ah,
because
yeah.
in a map around 17 disulfide linkage. So disulfide
Okay.
mapping also very important whenever you want to compare with the innovator versus your molecule.
Okay.
So sometimes during the analysis, that is it. And in secondary structure, we are doing the far UV and near UV analysis. That will determine the far UV will determine your secondary structure analysis, and near will tell you the tertiary structure analysis.
Okay.
In FLR, also we can use for the 280 and 295 estimation excitation range. with the emission spectra, and that will ensure that the tryptophan molecule which is present in the surface or in the buried conditions and the tertiary structure remains same like innovator molecule, that will be also carried out in that case. And also in addition to that, we are also doing FTIR,
Yeah.
because in case of the molecule amide 1, amide 2 is very important character. Amide 1 itself will tell you the alpha helix, beta helix and other structures which is present in the particular
Yeah,
protein.
thank you very much for this information. I almost seem like we need to have another discussion on the protein characterization. You listed seven or eight, or maybe more than a dozen different parameters to ensure that biosimilar is similar to the original molecule. So maybe we should have another separate discussion on that. It seems so
Sure,
interesting.
sure.
So Dr. Majumdar, the next thing which I wanted to find out from you is that so the Indian biotech industry, in terms of recombinant proteins and in terms of mAbs, have come long way in past 20 years or so, especially for biosimilars. So where do you see the industry will be in 10 years from now, 15 years from now? I mean, what are the challenges at this time for mAb industry?
Yes. See, when I was in Wockhardt, it was a year of 2004. In
Mm-hmm.
that case, the people have started working on maps, but not in full fledged. But
Mm-hmm.
now, if you see, every industry is working on maps. Those
Yeah.
are in, especially for biotech industry. I'm talking about Biocon, Lupin, Serum, and even in Wockhardt. So these people are really very good in biotech industries, and they
Mm-hmm.
are mostly focused on the biomolecule, and they are working on maps because
Mm-hmm.
see, this mAb is now very hot cake of the hot molecule, but
Mm-hmm.
the problem is that the price
Mm-hmm.
was very high. That's why that price was not comparable with the innovator. But now patent has expired, and the people start working on mAbs in a different type of mAbs, because the medical applications of the mAb, it is a kind of magic bullet. So it will
Mm-hmm.
act on a very specific direction to control that. And in India, it is now
Mm-hmm.
it is possible. But at the same time, when it is progressively elevate its own infrastructure for the map, because mammalian cell line maintain as compared to microbial cell line maintain is a big challenge,
Mm-hmm.
because there is a chance of contamination.
Okay.
There is a chance of contaminations, and although the life cycle of the fermentation's life cycle is 17 days or 15 to 14 days, but that every analytical parameter from the beginning to end till the fermented to DP, in every step that you have to integrate your analytical parameter and to ensure that the product whichever you are preparing, it is exactly similar to your innovator molecule.
Okay.
And in that case, that whenever at the same time, that is a product life cycle I'm talking about. So at
Mm-hmm.
that same time, when from the toxicology study and phase 1, phase 2, phase 3 market, they are available from the clone to DP till market authorization. Whenever you are submitting the author document, so document to the regulatory agency, now the requirement is very high. The requirement is
Mm-hmm.
not only the SDS page or any preliminary characterization step. They need in details characterization to ensure that product is not comparable. It is a similar to your innovator. So
Mm-hmm.
the judgment by using that regulatory agencies are very crucial nowadays. That I feel.
Okay, okay. So regulatory direction is going to be a important challenge in coming years to make sure that all the regulatory guidelines are met.
Yes.
Yeah. Okay. So, Dr. Majumdar, the next thing which I wanted to discuss with you is nowadays everything is based upon artificial intelligence. Everybody is talking about artificial intelligence. So, how do you see it is going to affect the biotech industry?
I will suggest that AI is a very powerful tool, and for the literature survey and the basic information to initiate the many development activity,
Mm-hmm.
it will shortage the time period than a normal part.
Mm-hmm.
But use the AI course is also very useful for the pharmaceutical industry, because nowadays AI will slowly grow in the industry, because every system also as a customer support they are utilizing the AI. So whenever we
Okay.
do the method development or anything, so
Mm-hmm.
analyst is asking the boss if it is not available, then some problem, then if the column is clogged, what to do, what not to do. So nowadays AI is giving the solutions.
Okay.
So on
Okay.
that basis, most of the thing is now hassle free. But don't depends on the AI, because molecule, you
Hmm.
know your molecule
Okay.
better than your AI.
So, Dr. Majumdar, the next thing that I wanted to discuss with you is the public perception about the biotechnology. There are a lot of information going on in general public about like how good the biotechnology is, because it involves the manipulation of DNA, it involves the manipulation of the whole genetic sequence, and people are more aware of it since the COVID times, and especially now with CRISPR technology that can allow lot of gene editing things inside the cell very quickly. Some people are more getting more concerned, especially in US, and I'm sure it is happening in India also. So I just wanted to get your feedback on it.
Yeah, in case of genetic manipulation, it doesn't mean that the alteration of your own genetic sequence. But sometimes what happen in case of mRNA vaccine, if you compare that mRNA vaccine which is integrated. So I went to Belgium and we have a collaboration with for the mRNA vaccines. So there is a MIDI machines and was there. So the mRNA was encapsulated, and that mRNA is integrating with the sequence to product that will follow the central dogma.
Hmm.
DNA to mRNA, mRNA to protein, like that. That this central dogma they will follow by the translation and transcriptions mechanism. And there the mutation was done because of the increase the productivity and more target oriented matter in that case. But definitely the original genetic manipulation of the sequence, if you compare with other protein like that, so that was a conversion, or that was a doubt that the original genetic sequence is going to impact. But there is the pathway is different.
Hmm.
Always try to understand the pathway is different, because whenever you are using a recombinant protein vaccine, it is very target specific. But whenever using a mRNA vaccine, that will be integrated, and it has observed that sometimes it is going to impact on the genetic manipulations, but it is not going to be too much harmful that when the as the people are expecting, or the people are thinking about it. So that
Hmm.
was my perception. It was not the correct idea.
Okay. 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 And so, the Dr. Majumdar, one thing which I also wanted to ask you is this particular podcast. I'm sure a lot of people who are younger, from younger generation, who are entering the biotechnology field after doing their bachelor's or masters, would be listening to this podcast. So I just wanted to get your feedback on, like, what do you suggest the younger generation, what field they need to focus on, what qualities they need to polish, and any advice for their success.
See, I would like to suggest that whenever you study that the biotechnology, so microbiology, biochemistry, and biotechnology, they are very correlated to each other. They are
Hmm.
mostly, they're very most of the subjects matter was the same. But thing is that if you have a good basic information should be very clear, because I have
Hmm.
observed whenever you are dealing with a protein, they don't know the amino acid structures. So this amino acid is the building block of a protein. So the basic root should be very strong, because industry has its own requirement. Some industries focused on the vaccine, some industries focus on the recombinant DNA protein, some industries focused on the synthetic peptides. But if you see that protein is the same, so basic knowledge or basic principle about the chromatography, about the protein sequence, about the protein generation, that biochemistry part, microbiology part, and technology, and how it is going to utilize in the pharmaceutical technology. So that thing should be very crystal clear in their mind, so that they can diversify their knowledge as per the industry requirement.
Hmm.
Whenever the any industry, because you don't know if Serum is suppose selecting some person, Serum Institute of India, they are more focused on vaccine. So they will tell you that you have to give some contribution for the vaccine. So that
Hmm.
time you do not say that I have a knowledge of biochemistry, I don't know vaccine. You
Hmm.
try to cultivate, diversify your knowledge towards the vaccines by
Hmm.
using the basic principle. If you are
Hmm.
joining INTAS, if you are joining USB, so that the same way, because field is see, think just is a protein. So whatever the basic information, basic principle. For example, we used to do during our post graduation study Nelson and Cox, Straya, Leninza. These are the basic principle, but people are using nowadays Google, because they are mostly familiar with the Google Perplexity and all these things. But basic study book should be very crystal clear.
Mm-hmm.
Till date, if any thing is required, see, if you are using nanodrop, on yesterday I was just thinking about that how to calculate, one, suppose one person was asking me how to calculate the concentration in nanodrop, because that will give you a one drop of sample. And so same Lambert Beard law, how to use it, and see, everywhere your basic principle is aligned, so you should be aligned and more concrete about your basic principle and crystal clear idea during your post graduation. It is very important.
Yeah, I think, but thank you for this advice. I think you said something which is very, very important. Keep your basics very strong and be ready to adapt to a new environment. I mean, but the basics will never change, whether it is vaccine industry, whether it is mAb, or whether any other related biotechnology field. As long as basic is strong, they
Yes.
will do very good. So that's a very good piece of advice. So, Dr. Majumdar, I think with this we are almost towards the end. And for the end part, I want to do two things. One is I request you to share a funny story from your biotech career, whether it is a bench work, whether it is interaction with the people, whether something funny during a conference, something like that.
I will take you one example that during this method development activity, it was a thing that we did RPH PLC and for the relative impurity analysis. So one guy did what thing, that he analyzed the sample in C18 column, and in
Uh-huh.
the C18 column, when they analyzed, he has got a very clear, crystal clear peak, and that peak position was 0 to 5 minutes period, and based on that only, and he has validated that method, and he has also submitted to some agencies. And this method was, in fact, it was a very funny thing that protein was inside the column.
Uh-huh.
It is only the buffer peak. It was only the buffer peak. So whenever we are using that, so that C18 column is captured, and suddenly this when the phase 3 trial was there, so same document they wanted to do with some scale up batches like
Mm-hmm.
2000 liter batches. So in
Mm-hmm.
that case, when we did the submissions, we see that it is only the buffer peak. So it was a, we just increase the acetylnitrite concentration, and that of this will give you a very good peak shape in a gradient, linear gradient way. So that time it was highly surprised, and people was funny that the based on the buffer peak also, even the reviewer also approved, and that process is gone now sequentially ahead. But the exactly the protein which is need to isolate or need to characterize or need to quantify, that was inside the column because of the strong binding affinity with the column ligand.
Oh, okay.
So
Okay.
that was a strong binding affinity with the column ligand. Actual hero was inside. Only
Mm-hmm.
the background was came out, and they
Yeah. Yeah.
realized that's the real hero. But that's real hero was not there.
Yeah.
So
I mean,
we
chromatography.
think,
Yeah, yeah. I'm
yeah,
sorry.
chromatography is a very very interesting part. So that's why I'm telling you that basic information, when because when we increase it acetyl concentration, that we come to know there's a huge number of protein which is inside the column, it is coming out. So
Yeah.
it was a really funny thing, because that was already approached to the regulatory agency. We take it back and then we revalidate the method, and these kind of things are happening because definitely that is people working on the pressure, but you should know that what actually you are doing. So
Mm-hmm.
these things is very careful while you are
Yeah.
submitting the documents.
Yeah. Chromatography seems very simple, but it's a very complicated science, and it is amazing. I mean,
Very complicated science.
yeah, I think if somebody shows the different columns, I mean different columns and different types of depending upon the size of the protein, they are coming down through the column with the different colors. It will be very impressive. I mean, for the general public, people who don't know about it. I mean, for example, chromatogram. I mean, why it is called chromatogram? Because it is colored. I mean, that's the reason it is called chromatograms. These are very, very funny things. Okay, thank you. So, Dr. Majumdar, now we are coming to the end. So we will end this podcast by asking or responding to rapid fire questions. So I'll ask you 10 different questions, and you need to give your response on a scale of 1 to 10. 10 being the best almost there or strongly agree, depending upon the question. And if you are not comfortable in answering any question for whatever reason, just say no answer. So I will start now, and our aim is to finish this in less than 60 seconds. So first question: quality of biotech drug products in India.
6,
Okay. Production capacity in India.
10.
Okay. Innovation in India. IP protection in India.
It's 6,
6. Adequate availability of talented workforce across key functions.
8.
Okay. Productive interaction among peers.
It's around 7.
7. Okay. Capital available to support and commercialize new ideas.
It's 6,
6. Cost efficiency with an aim to reduce the cost of drugs.
9.
Okay. Unambiguous guidelines for regulatory process.
9.
Okay. And finally, make in India efforts.
It's 9.
9. Okay. Thank you. Now, so this is the end of the rapid fire questions, and also this is the end of our discussion. So, Dr. Majumdar, thank you very much for giving so much of information about your journey and how biotechnology is moving, how it has moved in past 10, 15 years.
Thank you. Thank you very much, Dr. Arun.